Today's talk is a great listener. The beta cell primary cilium is an antenna for islet intrinsic and extrinsic signals. And we have Dr. Andreas Müller from the Ller lab at the Langerhans Institute in Dresden speaking to us regarding this topic. Just a little bit about him. Andreas Müller studied biology at TU Dresden. He got his PhD at the Langerhans Institute in Dresden at the medical faculty. And during this time, he developed a novel correlative light and electron microscopy, CLEM, workflow to study the turnover of insulin secretory granules in the pancreatic beta cells. For his PhD thesis, he received the Thesis Award from the German Society for Electron Microscopy, and during his postdoctoral studies, he focused on the intracellular transport of insulin secretory granules using FibSem to reconstruct the microtubule networks of beta cells and their interactions with insulin. Recently, he became interested in the structure of beta cell primary cilium and its role in pancreatic islet signaling. And we have his paper for you to take a peek at in the chat. And thank you so much, Andreas, for joining us.
Yeah, thank you, Monica, for the introduction and also for the opportunity now to present some of our recent work. And you can actually see in the title slide already a tiny primary cilium here on its way through the islet, and it's also connected to a little axon here, which is quite interesting, but I will get more into detail on this in the second part of the talk. I will just give you a brief introduction on cilia in general. And I guess you know we have some cells of our body have motile cilia, like sperm tails or lung epithelial cells. And a lot of cells in our body have these non-motile cilia, and those are usually called primary cilia. And these motile cilia are important for locomotion, for fluid flow, and also other things, even also for sensation. And cilia in general are microtubule-based organelles, and by this, they stick out of the cell and they form a specialized membrane compartment that has a distinct protein and also lipid composition. And in the motile cilia, for them to move, you have these 9 microtubule outer doublets that you can see here and the central pair. And then you need most of these so-called motility components. So these are motor proteins that need to work together to make this regular beating of the motile cilia. Now, in the primary cilia or non-motile cilia, we don't have the central pair. We also don't have many of these motor proteins as in the motile cilia, but we still have a lot of distinct membrane receptors on their membrane. So it's really like an antenna that can receive special signals from the cell exterior, and they are important in hedgehog signaling, for example, so during development, but also in fully developed cells for cyclic AMP signaling, for example, and calcium signaling. So our beta cells also have primary cilia, and we know this since the 60s, but in the recent years, there has been a lot of new data on their role actually in the islet. We know that they project from the avascular pole, so they are usually not connected to the blood vessels or not very close to the blood vessels. They are important for paracrine signaling in the islet, also because they present a lot of different receptors, somatostatin receptor, also the insulin receptor has been described, certain GABA receptors. And we know already from some mouse studies where the cilia have been destructed that dysfunction of cilia impairs insulin secretion and leads to a diabetic phenotype in mice. And there are also now more and more studies coming with genomic data showing that actually there is some dysregulation of ciliary genes in type 2 diabetes, for example. And also a recent study from the youth lab suggests that there is cilia movement in response to glucose. But what we are currently missing is really a complete 3D structure of the beta cell cilia and also a comprehensive mapping of their interactivity with other islet cells. And I think this would also help us to better understand cilia function within the islet. And for this study, we basically had 2 questions. So the first one was, since my focus is really more on structural biology, how is the cilium exoneme, so this microtubule structure, organized in 3 dimensions? And can this structure actually support an active movement that would be similar to motile cilia, for example? And the second question, which I think is also quite interesting, how do cilia interact with other islet cells, but also with the surrounding tissue? And to answer the first question, we used different kinds of so-called volume electron microscopy. And this is what you can see here. In panel A, you see FIP-SEM data, and this is basically a continuous electron microscopy stack at 4 nanometer voxel size. And you can see here in this panel the edge of one beta cell with the insulin granules here with some mitochondria, and in orange you see the primary cilium in cross-section. And when we look from the other angles, we also can see it then in the longitudinal section. And what we can see here already in these cross-sections, we can discriminate single microtubules and also these doublet microtubules. But of course, the resolution is a little bit borderline. So we wanted to also get some data from human cells, and there we used samples from our cohort of living donors. But for this, we didn't use the focused ion beam scanning electron microscopy, but this was done by series section electron tomography. And you can see here in this panel that actually we needed to stitch together 8 sections with smaller volumes to really get the full volume of these human cilia. And what you can see here now that we can better resolve these microtubules, because with this method we are a bit more flexible in choosing our resolution. And in the next slide, I want to show you one video of the FIP-SEM. So this is this continuous volume of the border between 2 cells. We start here and see these microvilli in between the cells. We see the insulin granules here. And when I start the video now, we will now go from the tip of the cilium, which you can see here with the single microtubules. Then we see these doublet microtubules closer to the base. We see the basal body here and also the daughter centriole next to it. And then we can move now into this 3D rendering of the microtubules and also of the membrane of the cilium. And just to mention this, these kind of reconstructions have been done by now only for a few cell types, so usually for kidney cilia. So this is actually the first to do it in primary cells. But we see similar things that we see also in the kidney cells. And this is also what we see now in the human here. And these are just some still images of the 3D rendering. What we see in general that this microtubule structure, this axoneme of the primary cilia, is much more disorganized as compared to motile cilia. So first of all, we don't have the central pair, and also we don't find it here. And we have an early termination of these A and B tubules. And usually these B tubules terminate a little bit earlier than the A tubules. What we also can see is this transition of some of these outer doublets, and they are at the base of the cilium, really on the edge of it. And then during the length of the cilium, they will transit towards the center of the cilium. And this is what you can also see here in this plot. So when we plot here the distance of the microtubules to the membrane of the cilium, we see that some of these doublets make this transition to the center of the cilium, but not all of them. But this is somehow a feature that has been seen also for these kidney cells. So then we were asking if these cilia actually have also these motility components that would allow them for an active movement. And for this, we used expansion microscopy. So this is now fluorescence microscopy, and it works by physically expanding your sample to gain then a higher resolution with a normal fluorescent light microscope. And this was done by our collaborators in Milan, by Nicolai Klener and Gaia Piccino. And this worked by cryosectioning of the pancreas, hydrogel embedding and denaturation. Then these pancreas slices were expanded and could be labeled with antibodies. And we compared here our beta cell cilia that you can see here in panel B and D with mouse tracheal epithelial cells. And these are motile cilia, right? You see here in this electron microscopy cross-section, the central pair. And you can also see some electron-dense spots, which would be these motility components. And we checked now different markers that are important for this active cilia beating. And we could find them really in regular patterns along these trachea epithelial cells, which you can see here in panel E. But we could not find a lot of signal in the beta cell cilia, which means that the beta cell cilia have classical features of these non-motile cilia, and it does not necessarily rule out that they move, but when they would move in a different way compared to motile cilia, right? They are not able to perform like a regular beating. So then I would like to move to the second question. How do beta cells here interact with other islet cells? And this, I think, can also tell us something about their way of signaling in paracrine signaling within the islet. And for this, we also looked into expansion microscopy to get some basic values of beta cell cilia interaction with the neighboring cells, also an idea on their size and abundance in the tissue. And what you can do with expansion, you can additionally stain your sample with an NHS ester, and this will give you almost an electron microscopy-like image of the background of the cell. And this is what you can see in panel A. So with these stainings already, only with the acetylated tubulin and this ester stainings, we can discriminate already the tissues, the tissue types in the pancreas. And we see here, for example, we can see the exocrine, we can see erythrocytes in the blood vessels, we can see the duct, and ductal cells have these quite long primary cilia, and we can also see the islet. Within the islet, we cannot really discriminate beta cells from alpha cells, but we can do this, of course, with an additional staining against insulin, and this is what you see here. So the insulin in green, acetylated tubulin as one of the markers for primary cilia, and also additionally ARL13B, which is a small GTPase that is also cilia specific. And we were looking first in the ciliation of the beta cells, and we got values around 80%, which is in line with what has been described in the literature. We could also measure the length of ductal cells and non-beta and beta cells, also data that are in line with what has been described before. And we were already starting to look in the number of, or in the percentage of contacts of beta cell cilia with beta cells, with non-beta cells, also with capillaries and with the acinar cells. And you see here that beta cell cilia mostly contact beta cells and the other endocrine cells, a little bit also capillaries, which is interesting because this has not been described in the polarity before. And they are also in contact with acinar cells on the edge of the islet. And now we wanted to look into these interactions more in detail in our electron microscopy data sets. And now for this, we mostly had data from isolated islets. So I will start now with the interaction of cilia with other islet cells. And what we could see is that there was, first of all, a pretty high variation of the length of the ciliary pockets. And this is what you see here. So some of these cilia were really almost internal in the cell with these long pockets, and then they leave the cell. And you will see in the 3D animation that this cilia actually is longer because we are now looking only at a slice. But what the message of this image and this slide is that these cilia are usually very restricted in space. So either by their ciliary pockets, and then when they leave the cell, also by the neighboring cells. And this is what you can see here. You see the long pocket, you see this, the cilium is out of the cell, and then it's in contact with 1, 2, and 3 cells. And there's also this cell here in the center. And we can take a quick look at a 3D animation of this particular cilium. So we have now the cell interior with the pocket, the cilium leaving the cell, and then it is in contact with one neighboring cell, and then there's the second one and the third one, and you see that it's totally squeezed in between these cells. But this also opens a lot of opportunity for interaction, right? So we see cilia that are only in contact with one cell, but we also see, like in this example, that cilia can be in contact with more islet cells in their vicinity. We also found some kind of even closer interactions, and it's not really easy how to call this. I called it pinching in the beginning, and what you can see here in panel A, and this is now human pancreas again, that this cilium here is really pushing the membrane of this neighboring alpha cell, which also would mean that there could be a lot of interaction going on, and we don't really know what the factors that are involved in this pushing, or if there are some adhesion molecules that help to make these contacts. And what we also saw occasionally, that beta cell cilia of neighboring cells are meeting in the extracellular space, so probably also to communicate. So this, what I showed you, was now on isolated islets. And in our volume EM data, we didn't have mouse material from whole pancreas. We had pancreas from the human, but this is hard to image in high volumes. And last year, we got access to a FIP-SEM mouse pancreas data set that was not generated by us, but by our colleagues at Genelia. And this now contains almost a whole islet in 3D, and I will show you some movies later. And here you see the exocrine tissue next to the islet. You can also see blood vessels. So we basically have all the cell types that are non-endocrine also here, and we can really look at the tissue interaction really at high resolution. And we were first interested in what is happening to the beta cell cilia on the edge of the islet. Do they have maybe a little more space as what you would see when you isolate the islets, or are they also squeezed into these narrow spaces even with the exocrine? Or maybe they also communicate somewhat with the exocrine tissue. And this is what you can see here. So you see a beta cell cilium with the basal body and the axoneme, and next to it an acinar cell with these really large and dense granules. And in the 3D reconstruction, you can see here that also this cilium is really squeezed in this narrow space next to the exocrine. We were then looking also into the interaction of cilia with blood vessels, and we could see this already in the expansion microscopy that we had some cilia like here stained with acetylated tubulin that were close to the blood vessels, and you can see some red blood cells here also in this case. And we could also see this in our electron microscopy data set, that in some only very few cases these cilia were contacting the blood vessels. So what was even more interesting and what I was very excited about, that in this data set we also see islet innervation. And I will now start this movie, and you will see on the bottom here the endocrine part, so the islet. These empty spaces here are the blood vessels, and on top you see the exocrine part. And we will now just go through this 3D volume, and I hope it doesn't run too bumpy now since this is online, but at some point we will see some nerve fibers appearing, and they are basically touching the islet. And you see here one, so you have several axons that are running along the beta cell here. And also here we have a second nerve fiber. And what we then found in these data, that we found cases where the cilium, and here we had the basal body and we have the cilium here, that actually the cilia were contacting these neurons, only very transiently, but we found also some evidence of presynapses on the side of the neurons. And now we move out of this data set, and I just want to highlight that this is really a 3D part of actually a much bigger data set. So again, we have here this nice tissue context, and when we can move to the side, we see that this is also an isotropic volume. So it's 8 nanometer voxels in all directions. And this is an open access data set, so anyone can have a look and also can cut out smaller portions of it with their cells of interest, for example. And what we see now in these still images in another context that we found in this data set, that we also find some small vesicles, synaptic vesicles probably, and we also see evidence of synaptic vesicle release on the cilium. We also find these interactions with alpha cell cilia. So you see here an orange cilium of an alpha cell that is contacting this axon here, and next to it is a beta cell that also is contacting this little nerve fiber here. So this happens somehow frequently in this data set, but at the time we only had this one data set available. And of course we know that this is all very descriptive for now. This phenomenon has so far been described only in the brain, and there is this so-called serotonergic axon ciliary synapse that also, since it is a special signaling compartment, it also has a special way to then alter chromatin accessibility. So we have now found this at least structurally in the islet cells, and we now wanted to look further also with fluorescence microscopy to define better if this can be a functional synapses or if this is basically just a coincidence. And for this we turn to fluorescence microscopy of mouse pancreas sections. And I just want to start with this one image of pancreas innervation in general. And I know, several groups have been working in the past on pancreas innervation. So we stained the axons with beta-3 tubulin, which is a general marker for neurons, and we used synapsin 1, which is a marker for the presynapses, for the synaptic terminals. And what you see here, mostly that in the islets, we have these axons that are then decorated with these synapsin 1 positive boutons. So these are en passant synapses that are connected to the islet cells, also to the blood vessels. And we were interested if these kind of connections are also happening for the beta cell cilia. And this is now what you can see here. So we found some of these axociliary synapses, and we also saw that they are positive for synapsin 1. So you see here in this panel, this large nerve that is splitting into single axons. In white, in magenta, you see again the synapsin 1, in green the insulin, and ARL13B is the specific marker for the primary cilia. And we found really some cases where the cilia were in very close proximity to the synapsin 1 positive signals, and this is what you can see here. So they are really making contact with these axons, and synapsin 1 is a marker of the functional presynapse. So there are probably most likely synaptic vesicles there that are then released towards the cilium. We found that around 12% of beta cell cilia contact these axons, and of these, 70% are synapsin 1 positive. We then wanted to look at this at even higher resolution, so we turned to structured illumination microscopy, which is a kind of super resolution microscopy. And here you don't see the nerves, but you see the synapsin 1 signal that is also close to the cilia here in cyan. So we could really see that the synapsin 1 nerve terminals are very close to some of the cilia. Now, synapsin 1 is a general marker, so it does not tell us yet the neurotransmitters that are involved. So next we screened for many different neurotransmitters actually, and we got a positive signal for acetylcholine, and specifically for the vesicular acetylcholine transporter, which transports the acetylcholine in the neuronal vesicles. And what you can see here is again staining against beta-3 tubulin, the nerves, ARL13B, which are the cilia, insulin, and vesicular acetylcholine transporter. And similar to the synapsin 1, we could detect here these signals close to the cilia. And actually 65% of these contacts were positive for acetylcholine. So we believe that in these axociliary synapses of beta cells, acetylcholine might be the major marker, major neurotransmitter that is released onto the beta cell cilium. And we could also resolve this at a higher resolution here with structured illumination microscopy. And what you also see that they are close to each other, but they never really directly co-localize. So there's always a certain small distance, which means that they are on distinct compartments, right? The acetylcholine is on the neuron side and the ARL13B is on the cilia side. We also checked for postsynaptic markers, because this is what you would expect in a synapse in the brain, that you have a presynapse with these presynaptic vesicles, then you have the postsynapse, which is the dendritic spine with postsynaptic density proteins. And we could not detect any of these, but I just want to quickly mention that it has been proposed before that primary cilia can act as a postsynaptic compartment on their own, because they share some structural and molecular features and similarities with the dendritic spine. So they are an isolated compartment with specialized receptors, and the dendritic spine has basically similar features, right? So we think that it's not really necessary for the cilium to have these postsynaptic markers or to have like proteins that can cluster the receptors, because the receptors are already clustered in the cilium itself. So to quickly summarize my talk, we started with the structure of the beta cell cilium, and we found that it is disorganized and similar to what has been described for other primary cilia and other cell types. We could find numerous cilia cell and also cilia-cilia interactions within the islet cells and also to the non-islet tissue. And we could find axociliary synapses that are cholinergic, connecting to the islet innervation, which basically means that cilia are compartments for receiving islet intrinsic, but also extrinsic signals. So there are still quite a lot of open questions. So we still would like to find out if these contacts are really functional and if they, for example, play a role maybe in the cephalic phase of insulin secretion. We would also like to investigate what makes the signaling pathway special compared to signaling through the plasma membrane. What happens downstream of this cilia signaling, and are the beta cells that contact neurons with their cilia structurally and functionally distinct from the other beta cells? So for this, we have now the opportunity to hire a postdoc for the next 2 years. So if you know someone who is interested, or if you are interested, please contact me via this email address here. And this position will really focus on the functional investigation of these axociliary synapses. Just to give you a link to the preprint, this is now still in the last days of peer review, so we hopefully have this online soon, also with the new data on the fluorescence. And you can find the preprint here, and you should check out Open Organelle, because on Open Organelle we have all our islet Fib-SEM data, and there's also the pancreas data set. So this is, I think, really valuable, and it makes sense to look into this. There's a lot still to explore with these new data. I would like to thank my lab, especially Michele, and also the colleagues that helped a lot now during the revision. Then, of course, this was a very fruitful collaboration with Gaia Picino and Nicolai Kleiner in Milan. We have this long-standing collaboration with Genelia and now Yale. So I'm very grateful for Harald, Song, and Shan for all their help. Also the collaborators here on campus and the funding agencies. And I'm very happy to take questions now.
Thank you, Andreas. This is such beautiful work. I mean, it's absolutely fantastic and meticulous. Oh, here's a question. Yes, this is.
Hi, this is Vera Kravitz.
Oh, hi, Vera.
Hi, I just thank you so much, Andreas. I love your work. So I just have a question. Do you ever combine this imaging with the functional calcium imaging data, or something? Yeah, so to sort of get an idea of whether we can overlay different functional beta alpha cell subpopulations with the available electron microscopy.
I mean, this would be very interesting. We didn't try this yet, but yeah, I agree. We need some better functional data to combine with the structural data. I mean, we will definitely try to do more live cell imaging now, also looking at these synaptic connections. But we haven't yet combined it really with calcium imaging, but this would be very interesting.
Thank you.
Great question. A lot of work to be done there. I had a question for you. So can you talk a little bit about the specialized lipid composition of the primary cilia? Right, they have a specialized composition of the SCFAs, sphingolipids, and the LPS. And where do they source most of that lipid?
Yeah, I'm not really an expert on this one. I think I don't really know the answer to this.
Oh yeah, well, no worries, of course. I just brought it up because at onset of type 1 diabetes, there is a loss of the single chain fatty acid producing bacteria, and single chain fatty acid can induce cilia formation. So you just wonder, you know, is there some correlation there, right? Decrease in those bacteria, decrease in SCFA, and some kind of alteration of the primary cilia phenotype and functionality. You just wonder about that, but you know, obviously it could go both ways. So who knows?
Yeah, I mean, in general, what we are still also missing is like really the full view of you know, the protein composition, the lipid composition. We don't have that yet. I think we have now a relatively good idea on some receptors, for example, but I think it makes sense to look in the future also into like a full proteomic and lipidomic approach for the cilia.
Yeah.
Just to have an idea what to look for and what factors are interesting and are really there. To
fully characterize that landscape. And then in humans, like, does the density of the primary cilia change across the islet and then across the pancreas? Have you observed that yet?
So for our data, we only have now these snapshots because for electron microscopy, the problem is always the quality of the samples, and we are lucky that we found some good samples. I think Olaf Idewald in their recent paper, they at least show that the abundance of cilia in general compared to mouse is a little bit less. And I think even there are some papers that have shown this before. They could also show that the cilia are shorter in type 2 diabetes. Yeah, but our data, we really, our view is now too narrow for these kind of quantifications, unfortunately.
Yeah, it would be interesting to tap into the, pre-existing data sets where some data may just be sort of hiding out and bring those all together and maybe even use AI machine learning to kind of interpret them or create a larger picture of what's happening. I wanted to talk a little tiny bit about ciliopathies and ciliopathies, like for instance, the syndromes, Bardet-Biedl syndrome is associated with RA and type 1 diabetes, autoimmune diseases. What about looking into some of those samples just to kind of see, is there a repository for that disease state where you could kind of look?
I think there is, because in the UK there are also these regular meetings, and this is really more focused like on the cilia side, right? I think that there would be probably access to some of these samples, yeah.
Yeah, that might be fun. And then I guess I would say, another question I had was the trafficking of the ciliary membrane proteins, right, by the interflagellar transport machinery. Has anyone done any work to see whether or not the CVB, like the Coxsackie virus B, obviously we know it impacts the beta cells, protein synthesis, ER to Golgi trafficking, but does that other trafficking site also get modified by virus like CVB?
You mean that the cilium in general could be affected by the infection also?
Yeah, the trafficking of the cilia membrane proteins in their interflagellar transport system. Or is that like totally separate and not going to be affected? I'm just curious.
Well, all the proteins are transported from the Golgi, so I think no one has really looked into this. We also don't really know how dynamic the IFT actually is in the beta cells, because we know it's there, right? Because if you knock down IFT88, you don't have cilia anymore, also in the islet. In electron microscopy, we don't see a lot of these IFT packages, also in fluorescence, but it still might be very dynamic. But these are all things that we would need to look into. Yeah.
Well, the primary cilia is definitely having a moment, right? I mean, Jing Hughes and Jeff Millman are driving the charge over at WashU and your group as well. And, Matt Marins is sort of diving in there. So you've got a lot of people kind of looking at this area, and it's very exciting. I guess the one last thing I would say is, this is sort of in a sidebar, but, in the Bardet-Biedl syndrome, right? They have the association with autoimmune diseases. It turns out some of their BCRs, the B cell receptors, don't develop correctly. And they have this BB-some some of the components that make up the primary cilium are also present at the T lymphocyte and again, apparently the B cell synapse. So, how might you kind of further that, looking at maybe from your standpoint, looking at it, the B cell, the BCR, the T cell, the T lymphocyte, structures and whether or not they're missing some components, and maybe the cilia are missing some components. How might you do that?
You mean now more in the context of type 1 diabetes, or?
Yeah, type 1. I mean, genetically, you could look to see if there's some problems with those components, but I mean, structurally.
Well, ideally, what I think a lot of people would like to do is cryo-electron microscopy of islet cell cilia, but this is really hard to do.
Mhm.
I think it would be worth it, and also to combine it with proteomics approaches, because you can do, you know, this proximity labeling proteomics,
Mhm.
basically pull down the proteins of the cilia, and then also use this maybe as a starting point to look more into the structure, more targeted. I think this would be valuable to do, like in general. And yeah, I mean, cryo-electron microscopy would be great, but it's hard to isolate the cilia. Also in the primary cells, you cannot really enrich or anything. There are some ways to do it. We also looked into this a little bit, but so far this has been simply too time-consuming and tedious to really get a good amount of samples.
Mm. Yeah, it's just such an interesting field, and you wonder, you know, how you can kind of tease apart all these pieces. There's a lot of moving parts. It's not just, you know, it's a lot of moving parts, and now the interaction with the neuronal piece too is really fascinating. Okay, one last question. Thank you for the wonderful talk one more time. Just curious, which software did you use for the reconstruction?
So for the segmentation of the data, we use different tools, also some deep learning. We have a protocol for this, in Nature Protocols from this year. Actually, for the cilia, I mostly use like free software tools. So Elastic, iMod is the software that you usually for electron microscopy. And then for the visualization, I use either Dragonfly, which is also a software that has a free academic license, or Blender for this title slide that I had, which allows for a bit of nicer 3D animations. But for the segmentation, we tried to compile really the methods that we use in this Nature Protocols paper. So this is also, you know, with the people from our lab, with Michele Solimena and a friend of mine, Martin Weigert. So I think you can find this. Otherwise, I can also try to provide a link for this.
Thank you again for this beautiful contribution to the understanding of the primary cilia in context of beta cells and also type 1 diabetes ultimately. And we can't wait to see the full paper come out. We'll be looking for it.
Thank you.
Thanks again, Andreas. Have a great rest of your day.
You too.
All right, bye-bye.